And that's Passerina Cyania bringing us in, the indigo bunting. Welcome to the Dissectify podcast, the show that gets you curious about the wild world right outside your window. I'm your host, Nicholas Sines, bringing you content late every Tuesday night. And since it's so close to a holiday, I'm going to be doing this without video this time. And we're just going to be talking about whatever the hell was on my mind that's vaguely related to biology, which usually is too much. So today I thought I'd cover artificial selection and GMOs and synthetic biology, basically the things that people do in order to try to get living things to be more useful to us. Alright, so I always thought it was interesting that there's this really big distinction, this almost phobia between what people are comfortable with as far as changing living things in order to be more useful to us, right? We have this long history as humans of making, artificially selecting animals that are, well, not just animals, anything. Artificially selecting anything that's going to be food or is going to make some useful substance for us, right? So this includes animals that we want the meat of, we want them to grow up fast, we want them to be non-aggressive, we just basically want them to eat the things that we don't want to eat, grow up fast, and give me a whole lot of meat that I can eat, right? And I think that's pretty much the same for all food items, even plants. So in that case, we select things to produce a whole lot of the food item that we want reliably and all at once. We select flowers to bloom all at once and produce as big of blooms as possible, as bright as colored as possible. And in the wild, right, things don't follow those rules. They're going to be much more aggressive. They're going to try to protect themselves. They're not going to be as comfortable around humans. They're not going to get as fat as quickly. Basically, they're meant to survive instead of just being a tool to feed us. And we put a lot of resources into trying to figure out how to make these organisms produce food for us or services for us, right? We have horses and other livestock to pull equipment and take us places, and food items that are supposed to grow up reliably and quickly. And all of it was basically done with artificial selection. We found one creature that we're like, oh yeah, this one doesn't try to bite me or kick me or scratch me or whatever as much. And I like that. That's a feature that I want. So we're going to take that animal, breed it with another animal. And depending on the goal, right, you just kind of start refining down. So you breed the plant that produces the bigger corn cob or the bigger berries or whatever else. And you take those and you take the ones that have the traits that you want, breed them together, and slowly they become more of the thing that you want. And usually that's more of a food, more of a service, easier to deal with, more reliable, whatever it is. We're still doing that, and just at a larger scale. But with technology advancing, we've gone from that to discovering DNA and figuring out, oh, if we know what the genes can do, then we can take the gene from something and put it into something else, get the trait. So that seems to be a dividing line for a lot of people. And then there's this bad history with it because there's a whole lot of historical deceit, I guess, wrapped up in it. That combined with people just being afraid of something they're not aware of, right? I think it makes sense to be really careful with what you're eating, right? That's going to affect you 100%. Makes sense. If you don't understand something artificial, you're probably thinking, maybe I don't want to eat that. But I think there's varying degrees of this, right? And it has such a bad history because the company, I think, to my knowledge, is basically the only one that, or at least the one that had the biggest negative impact on it all, was Monsanto, which had the genius idea of taking this. Well, plants are hormone based. I guess let's start there, right? So they were taking this substance that kills plants, I believe, and this may be wrong. Maybe I'll look this up before I start spouting stuff off. Okay, so I'm probably not gonna edit this shit at all. So you're just gonna have to deal with the big silence. Well, I'll just have to deal with losing listeners to the big silence, I guess. Um, so yeah, I looked it up. Glyphosate kills plants by removing an essential pathway. So it blocks this pathway for making essential amino acids and um, removes the ability for them to make essential amino acids that are needed for growth. So basically, the plant can't grow. So they found this pathway that they could block, and they found a substance that could block it. And Monsanto had this genius idea. We're like, whoa, okay, well, what if we took this substance, glyphosate, and we know that we found a gene in a bacteria that's become resistant to glyphosate. We're going to take that gene from the bacteria and put it in the plants. And so all of the crop plants will have this glyphosate resistant gene, and they'll be fine. You can douse them in the stuff, this liquid glyphosate, and they won't be affected. But every other plant around is going to not be able to grow and going to die. So they were thinking, okay, well, this is really great, right? We don't want weeds around our crops, so we'll just genetically engineer, artificially put this gene in the seeds for the plants that we want to grow, the crops, and then plant those seeds, douse the fields in glyphosate. So anything else that's touching that soil, any plants that get in there and try to grow, are just not going to grow. They're going to die. And your crops are going to be fine. Well, as you can imagine, there were some issues. Not only were there some poor business practices and basically corporate bullying, but yeah, I don't think all the financial stuff's really worth getting into. Maybe it is. Why not? Fuck it. I want to talk about it. So you have these seeds, these genetically engineered seeds, and it works, right? Biology's messy, and you found a way to kill everything else. Okay, so seems like has potential, right? So they do this, but not only was it genetic engineering, but when people are already kind of nervous or afraid of things that are artificial, especially when it comes to food, and but yeah, Monsanto had this whole thing where they came up with these seeds and had it in the contract saying that if you buy our seeds, opening the package is like a legally binding document that says you aren't allowed to resell those seeds to other people. And you're not allowed to... Yeah, basically, they're just trying to control these seeds, and so it became this thing where if other farmers who didn't want to use these seeds ended up getting like some plants that ended up being pollinated or potentially getting that gene, or some stray seeds that end up in their field. And if that happens, then Monsanto is able to come after these people saying you're using our product and you didn't pay for it. So yeah, it was just a bunch of shitty business practices. And on top of that, dumping glyphosate into the soil ended up causing so yeah, I guess it's subject to an ongoing scientific and regulatory debate, it says. But there's this correlation of people who are around glyphosate a lot getting a specific kind of cancer, and that these correlations were found in the studies that the company did, but they didn't publish it, and it seems like they kind of tried to cover it up. So there's this kind of this perfect storm, right? These things that people already aren't excited about were coupled with these shitty business practices, and on top of that, potentially this really dangerous situation where these chemicals are making people sick. So, yeah, just not a good look. But from the biology perspective, I think GMOs are really useful. And they don't all have to be, right, this like environment destroying, cancer causing sort of a thing. I will say, I think biology is messy enough that a lot of people are, you have to be careful with these things, right? Because if you put a gene out there, there's a chance that, it has some effects that you didn't realize. It's going to interact with something else that's, cause some issues with the bugs or whatever, right? It might end up getting released into the environment, meaning like a plant is able to hybridize or something, and then now, oops, you just released that gene in like an environment that wasn't there before. So I don't know, maybe there is an argument there for GMOs being, I wouldn't say too dangerous, but too risky, potentially. Because if you're artificially selecting something, you're introducing not the same genes, but, right, mutations happen. So you're getting these new combinations and new functions or traits for some organism by happenstance, and then you find that trait, you think it's useful, you artificially select it forward, and now all the individuals in some line have it, right? But the difference is that can take a long time, depending on how long the generation time is between these organisms. Yeah, like if you wanted to artificially select elephants, that would take a long time. But the same is true with plants and stuff like that. That you find a trait, a lot of the times you're not going to get some new trait or something meaningful, or it may not be as heritable as you thought, and you're just kind of taking a shot in the dark to try to get it to show up in other plants, and then bringing it forward, you don't know how it, if it's heterozygous, meaning, it shows up when one version of the trait is there, right? Because you have two copies of each gene, potentially. So yeah, it just gets messy. And then, so in order to cut through all of that, you can do genetic engineering. Not only is it more effective because you know what it is and you know what you're trying to get it to do, and you can actually put it into a plant and say, this is what I want, and then does it happen? You're not trying to mess with all this messy stuff, seeing if there's traits that pop up that you can then continue. That's There's a lot more potential for it to be like a surefire thing. But you still don't know like what the impact is going to be. You still got to test it out and see if the plant is, the plant or whatever else is working out the way you want. It may cause issues in the organism, right? So that's not like a big ethical concern in plants, but in animals, right? That could be a big ethical concern. Yeah, I'm not sure if anybody's really trying to genetically engineer animals. I think it's mainly just plants for crops and, ornamental stuff. Like they released, I think it was like Firefly, this company made a petunia, but its petals glow in the dark because it's got some, I think it might be a gene from the firefly. Anyways, it's got this extra biochemical pathway that goes on in its petals, so that they can like intake sunlight and it'll like charge up this molecule that'll cause that tissue to glow, which is really cool. I bought one. It was like a little too expensive, maybe like $30, but it was a cool gift. And I think it was pretty impressive. It wasn't like glowing super bright, but it was pretty cool. So I guess what I'm trying to get at is people seem fine with artificial selection. In fact, we've done that for thousands of years for different organisms like broccoli, kale, cabbage, and cauliflower and kohlrabi. I don't think I'm missing one, but all of these vegetables came from the same mustard family. I think it was called, let's see, broccoli, and cabbage are what species? Brassica oleracea. So, brassicas are like these really tall, skinny plants. They came from wild mustard. And Brassicaceae are plants that are kind of weedy, I guess, is what people would think of them. They're like these really tall, skinny, can be tall. Most of the time I've seen them, they're like a foot tall, but skinny little plants like that with narrow leaves, and the flowers are pretty small and they have 4 petals, and that's it. That's kind of the telltale sign of a brassica member. But all of these, sorry if that's kind of loud, but all of these different crops came from selecting for the growth of different parts of this wild mustard species, right? I don't know how long it took, but I presume hundreds, maybe thousands. I don't know. Anyways, those are all results of artificial selection of different parts of this wild mustard species. And now we can do genetic modification, which is potentially a lot more dramatic. Well, for sure a lot more dramatic in that you're not getting, right, you can get a fairly large change basically overnight, relatively speaking, because you're putting the gene directly into the organism. But funny enough, I don't know, maybe I should check the NDA, but we are moving past genetic engineering in the sense that we are let's see. We're moving to a step that's more technically involved than genetic engineering, right? I think artificial selection and genetic engineering aren't going anywhere. Those are going to be there. They're tools. They're useful. They help us eat and all that sort of stuff, right? So they're not going anywhere, but we are kind of entering now an era of synthetic biology. I've worked personally at a company that printed DNA. Like literally they have a technology that they can put in DNA nucleotides and electricity, and I'm sure there's an error rate. I don't know what it is. I wasn't that high up in the company or that involved. I was just one of the pawns, one of the lab rats who, anyways. But yeah, their technology was somebody would put in a request, like researchers or companies put in a request for a DNA strand that they wanted, and it didn't even necessarily have to be an existing strand of DNA, although I don't think we're there to really be like saying, oh, we want this exact strand of DNA that's completely new, although there's an interesting point there. I'm probably going to circle back to a lot of this in another episode, but today is very much like, here's a topic, and I just kind of wanted to brain dump about it, I guess. So I worked at this company, and that's what they would do. Somebody would send in a request, hey, we want this sequence of DNA, presumably because they're going to try to put it into bacteria or a plant or something to produce some product or to study something, to study the effect of that thing, of that DNA on the organism, or to try to get some product out of an organism like a plant or a bacteria. I think that's what most of that research usually is. And I think that was most of the customers, was researchers. But yeah, they would put in a request and it would get printed, and then it would go through a process to where it would be sent off in a little vial, right? Like a cold pack that contains the DNA. They can continue their research. They have the little clump of DNA they can pull from. And so yeah, that was kind of eye-opening. I was excited to work there at the time. Not that I'm not excited about the technology. I don't even think it's inherently bad, right? I think there's a lot of risks, but I think there's also potentially a lot of benefits. But yeah, we are like moving from a world where it was artificial selection, being like, ooh, that tree has slightly bigger apples, and I want apples that are bigger, so I'm gonna, select for that and get apple trees that make bigger apples. Artificial selection. And then we move through genetic engineering, which is, I think this gene does this thing, and I want to test it out. I want a tree with bigger apples. I'm pretty sure this gene from somewhere else can help me from some other organism, can maybe help me get more, make that tree that makes bigger apples. So I'm going to put the gene in, grow the tree, see what happens. Yeah, now we can, right, like printing the DNA is, I guess, it doesn't necessarily mean a whole lot. It's kind of like one of the steps that would, a tool that in the future could allow completely new DNA to be like created and written off. Because before we just had to like find the DNA, get the DNA, move the DNA, right? Put it into something else. Now we can actually make completely new DNA, but I was thinking like, oh, well, we don't really have the technology to be knowing what the DNA does. Sure, we could like print off some random DNA, but it's not going to do anything. It's just going to probably code for some protein that's like non-functional, doesn't do anything, or just causes some issues or something like that. But the other day I was watching this Instagram, and they covered this paper. Yeah, what was it? It was like they made a brand new, right? A virus is not a living thing, technically. It utilizes the like machinery of actual cells in order to reproduce. So viruses do reproduce, but they couldn't do that on their own. They need a cell to get into to use the machinery to then replicate itself. And that's how viruses sort of propagate. And there was a paper, let's see, 2025. I took a picture of it, let's see. But what I did see of it was that they decided to do this in bacteriophage. So, which is a smart thing to do, right? You don't want to be making brand new viruses and then, see. Let's see. Generative design of So this paper is called Generative Design of Novel Bacteriophages with Genome Language Models. So, kind of scary shit, I think. Not because this exact thing is scary, but it really shows that we're on the road towards potential issues. So, remember how I was saying, we have artificial selection and then genetic engineering, and now we can print DNA, but we wouldn't really know what to do with it, was what I was saying. But this paper was released September 17th, 2025. And basically it's saying that they trained an AI model on a bunch of viral genome sequences and then said, we want a bacteriophage, so a virus that attacks and grows from bacterial cells. This is a scientist talking, telling the AI model, we want a brand new bacteriophage genome. And so let's see, maybe we should just read off the abstract first. It says, many important biological functions arise not from single genes, but from complex interactions encoded by entire genomes. Right, so biological functions come from multiple genes and their products interacting. Genome language models have emerged as a promising strategy for designing biological systems, but their ability to generate functional sequences at the scale of whole genomes has remained untested. Here we report the first generative design of viable bacteriophage genomes. We leveraged frontier genome language models EVO 1 and 2, those are the models' names, to generate whole genome sequences with realistic genetic architecture and desirable host tropism. That's a new one on me. So a host tropism is a pathogen-specific preference for infecting certain hosts or cell types. So all right, so they say using the lytic phage, what is that, sigma? Omega? I don't even know. Omega? Phi? I don't know. Anyways. X174 is our design template. Experimental testing of AI-generated genomes yielded 16 viable phages with substantial evolutionary novelty. Cryo-electron microscopy revealed one of the generated phages utilizes an evolutionary distant DNA packaging protein within its capsid. Multiple phages demonstrate higher fitness than the X174 in growth competitions and in their lysis kinetics. So they grow faster and they lyse cells faster. A cocktail of generated phages rapidly overcomes the X174 resistances, so the E. coli are resistant to this bacteriophage somewhat. They can resist the growth of this phage. And they're saying this phage, these phages that we made sort of using this one as a template, it performed better against these E. coli cells. demonstrating the potential utility of our approach for designing phage therapies against rapidly evolving bacterial pathogens. This work provides a blueprint for the design of the diverse synthetic bacteriophages and more broadly lays a foundation for the See, and this is the part that's, I think, scary. Lays a foundation for the generative design of useful living systems at the genome scale. And I think it's important to stress that, like, this is really baby steps, right? So bacteriophage genome size for X174. So about 5.4 kilobases, so 5,400-ish nucleotides. So really tiny genome. But they openly say this lays the foundation for the generative design of useful living systems at the genome scale. And we're a long way off because, humans, let's see, how long is human genome? 3.2 billion base pairs. So 3.2 billion nucleotides. That's a lot longer than 5,400. But this is like the first baby step in using, right, an AI system that can like find patterns, kind of have a rough idea of what a gene might do or where a gene is, and then like shuffle things and test them out and apparently come out with something that's like functional, which is kind of spooky. So kind of seems like we are starting to get all the bits and pieces we need in order to really make new things or shuffle things together, and then potentially at some point having the understanding to say, we want this thing, make it, and then it's going to be like a brand new living thing in the world that potentially doesn't have any of the well, I'm sure that would be really hard to do. It'd be hard, but I was about to say a living thing that doesn't have any natural like predators or whatever else, right? That would be the perfect crop. If you could make a living thing that grows fast, produces a whole lot of food, and didn't even need pesticides, right? Nothing could eat it. It has whatever has some insecticide, which sounds scary, but a lot of plants already produce insecticides. But it like produce some insecticide that like no insect in the world could handle or something. Which sounds great. But I think that's like another level. Because I don't even know what you would do if there was like an invasive species that... Because it sounds like the perfect way to cause like a serious invasive species. Because that's sort of what happens is you take this plant from somewhere else in the world, like, something from Europe or Africa or wherever else, and then like plant it here, and then oops, it like likes the conditions here. And not only that, like it can grow the plant, this hypothetical plant, but, or even animals or whatever, it comes here, likes the conditions, it can thrive, and it can thrive not only because the conditions are good, but it doesn't have all of those like predators or pests or things that would naturally like take it down a notch and keep it from taking over everything. Like invasive species don't have that. That's why they take over. And here we are talking about the first baby steps towards making artificial genomes. So I guess I'll talk more on this later when I'm more informed and it's not 2 a.m. Before Christmas. 2 a.m. on Christmas Eve day. Eve, I think. Anyways, these are some thoughts, and there's a whole lot of other stuff, I think, to talk about there, but yeah. Seems like we're working our way up the ladder. Artificial selection, genetic engineering, and now synthetic biology. And yeah, synthetic genomes. So, worth a thought. Hope you guys are having a good holiday. I had to circle back and record this because I almost forgot the most important part. Please leave a review wherever you're listening. Visit dissectify.com for more nature exploring tools. Not that I included any this episode. My Instagram at nicholas.signs can also be found there. Tag me in your nature photos. I'd love to see them. And remember, you live in a world with so many other species, so get out there and check it out. Thanks. Bye.